Hello, and welcome to the anatomy chapter. In this first part of the module, we're going to go through and talk about how we actually study the brain. So we're going to talk about different research techniques. A lot of them involve different types of imaging techniques that we're going to use and scientists use to study the brain. So the question we're interested in this first part of the module is, how do we relate brain regions to behavior? Now, one way is we can examine the effects of brain damage in both animals and humans. We can examine the effects of stimulating a specific brain area. We can record brain activity during behavior. And we can correlate anatomy with behavior. Now, this first part of the module is going to have a page for each one of these different ways we can relate brain regions to behavior. We're going to start off talking about the effects of brain damage. Now, when we talk about brain damage, specifically in humans, there's different types of brain damage that we might come across. First, there's acquired brain injury. So think about what this might entail. What would be a type of brain injury we would acquire versus a traumatic brain injury? So an acquired brain injury would be something like a stroke. And that's what I'm showing you over here in this image. You can see here's a blood clot, and basically what happens during a stroke is that you either have a blood clot that stops the flow of blood, and thus the surrounding tissue isn't receiving nutrients and oxygen, and cells start to die, or you have an aneurysm where you have a rupture of that blood vessel. We could have tumors. A tumor would be a type of acquired brain injury. Tumors in various parts of the brain are going to cause different types of behavioral effects. And then diseases like Parkinson's, Alzheimer's, Huntington's disease, all of these are different types of diseases we'll talk about later in the semester. But if somebody acquires one of these diseases, we can look at the brain damage that results from that disease and see how that has affected their behavior. There's also something called traumatic brain injury, or TBI. So an acquired brain injury is something that just happens to you over time for whatever reason. A traumatic brain injury is something that actually physically affects your brain. It's an external type of injury, whether that's you're playing soccer and you fall and you get a concussion. That could be a traumatic brain injury. You're in a car accident and you hit your head because you weren't wearing your seatbelt. All of those could be traumatic brain injuries. Now, when someone has an external injury, again, we can look at the type of injury that's occurred in the brain, and we can look at the effects on their behavior. Now, HM is an example of this, and you can click on the link to watch this video further on down in the page. Now, we can also look at the effects of brain damage in animals, and the difference between humans and animals is that in humans we have to wait for that damage to occur. In animals, we can actually do research and develop studies where we damage their brains on purpose, and that's a lot of what my research was about. So in the lab that I was in USC, we had a couple different techniques we used, and one was an ablation, which is a removal of a brain area. It's actually removing an entire brain area. And so I didn't do as much of this, but one of the focuses of the lab was to look at plasticity responses. And so one model we used was the motor cortex. And so one hemisphere of the motor cortex would be removed, and then we weren't really looking at the physical plasticity or the behavioral plasticity response. We were looking at what happened in the motor cortex of the other hemisphere to allow that recovery to happen. But we would actually do a surgery where you open the scalp, remove the skull, we would suction out a part of that motor cortex, then we would put the skull back on, staple up the scalp, and let that animal recover. Another type of surgery that's done is called a lesion, and this is where instead of removing the brain area, you damage the brain area. And this is a much more realistic version of what brain damage in humans looks like, because when someone has a stroke, that area isn't removed. That dead and dying tissue remains there and can cause further problems. And so we also, when I first came to the lab, they did a lot of the ablations, and then as my time there progressed, we started doing more lesions, where instead of removing that tissue, we would either go in there and kind of scramble it up so that we would be killing cells with a scalpel, or one model was using a little heating element to kind of scorch the cells, and in that way they would die. And so it was, again, mimicking much more of a stroke than just removing that brain area. And again, we were interested in looking at the plasticity response, the proteins and the genes that we saw that occurred with that recovery. Now, in order to do these types of surgeries, we use what's called a stereotaxic instrument. And so here we see it here, and the nose of the animal, we use mice and rats, would go here. This would hold their ears. And during the surgery, the animal's anesthetized, so they're not feeling anything. When they wake up, they act just normal. They don't seem to be in any pain or anything like that. And so you can use this to do the surgeries like we did, implant electrodes into the brain. We've done all sorts of different things, and this stereotaxic instrument is what's used. And what's kind of interesting about it is that these, on these little doodads, for less of a technical term I have, are measurements. And so you can use a rat brain or mouse brain atlas and get very specific measurements of what you are damaging in that animal's brain.